Good morning everyone, especially to ma'am, good morning. Our topic for today is all about molecular regulation of enzyme. But before I start, I want to ask first, what is enzyme? Okay, so enzyme, it is a protein that works to assist a chemical reaction by building or breaking down molecules. An enzyme can be regulated in ways that either promote or reduce their activity. And always remember that there are different kinds of molecules that inhibit or promote enzyme function. Actually, many enzymes don't work optimally or even at all unless bound to a specific non-protein helper molecules or ionic or hydrogen bonds. Now, let us know kung ano ba yung two types of helper molecules. So, the two types of helper molecules are cofactors and a coenzyme. Binding to these molecules promotes optimal conformation and function for their respective enzyme. Now, what is cofactors? So, cofactors are inorganic ions such as iron and magnesium. A cofactor is a non-protein chemical compound. Okay, non-protein chemical compound or metallic ions that is required for the enzyme role as a catalyst. Actually, cofactors can be considered as helper molecules that assist biochemical transformation. Next is coenzyme. So, ano nga ba yung coenzyme? Coenzyme are organic helper molecules. It is organic helper molecules with a basic atomic structure made up of carbon and hydrogen, which are required for enzyme action. A coenzyme is defined as an organic molecule that binds to the active sites of certain enzymes to assist in the catalysis of a reaction. Now, most common sources are dietary vitamins and coenzyme. So, an important step in the breakdown of glucose to yield energy is catalysis by a multi-enzyme complex called pyruvate dehydrogenase. So, ano nga ba ang pyruvate dehydrogenase? So, pyruvate dehydrogenase is a complex of several enzymes that actually requires one cofactor or a magnesium ion and the 5 different organic coenzymes to catalyze its specific chemical reaction. Therefore, enzyme function is in part regulated by an abundance of various cofactors and coenzymes, which are supplied primarily by the diet of most organisms. Actually, vitamins are required for enzyme to function properly. And multivitamin capsule usually contain mixture of all the vitamins at different percentages. Now, ano ba yung difference between cofactors and yung coenzyme? So, like what I've said kanina, yung coenzymes is organic molecules, while yung cofactor naman is inorganic ions. Okay, a cofactor is a non-protein chemical compound, while a coenzyme is a non-protein molecule. Now, let us move to induce fit and enzyme function. So, for many years, scientists thought that enzyme substrate binding took place in a simple lock and key fashion. This model asserted that the enzyme and substrate fit together perfectly in one instantaneous step. So, let us imagine that yung enzyme natin, yun yung ating lock, and yung ating substrate naman, yun yung ating key. Now, the induce fit model expands upon a lock and the key model by describing a more dynamic interaction between the enzyme and the substrate. As the enzyme and substrate come together, their interaction causes a mild shift in the enzyme structure that confirms an ideal binding arrangement between the enzyme and the transition state of the substrate. And for us to know better what is induced fit and enzyme function, let us watch this video.
Enzymes are protein molecules that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, is the area where enzyme activity takes place. An enzyme works by binding to molecules, called substrates, at its active site. The job of the enzyme is to convert the substrate into a different product or products through a series of chemical reactions. Following the reactions, the products are released and the enzyme is free to act on another substrate. At the active site, the substrate should fit into the enzyme almost like a key fits into a lock. Because of this, most enzymes can fit only one substrate. To accomplish this lock and key fit, the active site undergoes a slight change in shape in order to better accommodate the substrate. This is called the induced fit model because the enzyme is induced to undergo small changes so that the substrate can achieve optimum fit. All enzymes have optimal environmental conditions that are correlated with maximum enzyme activity. Environmental conditions such as pH and temperature play a role in how efficient the enzyme is in conducting its reactions. When conditions are less than optimal, an enzyme will lose its configuration and slow its activity. This is due to a change in the three-dimensional shape of the enzyme and is called denaturation.
So that is induced fit and enzyme function. Now let us move to enzyme-substrate complex. So the chemical reactants to which an enzyme binds are the enzyme substrates. There may be one or more substrates depending on the particular chemical reaction. So in some reaction, a single reactant substrate is broken down into multiple products. So the location within the enzyme where the substrate binds is called the enzyme's active site. The active site is where the action happens. So the fact that active sites are so perfectly suited to provide specific environmental condition also means that they are subject to influences by the local environment. It is true that increasing the environmental temperature generally increases reaction rates and enzyme catalyzed. So for us to better understand what is enzyme substrate complex, let us watch another video.
Substrate molecules bind the active site of an enzyme molecule. After the enzyme-substrate complex has formed, catalysis can occur, followed by the release of the product or products of the reaction.
So that's all, and thank you.